Hello and welcome to Effortless Mastery. Today, we are going to be doing a quick review of our last class on chemistry of nucleotides, chemistry of nucleic acids. What are those questions that can easily come to you in a medical exams, in a paramedic exams? What are those questions to see? What are the things to note about nucleic acids? Now, as we had learned that nucleoproteins contain that nucleic acid are usually in the form of nucleoproteins, and when they are broken down into nucleic acids and proteins. The nucleic acid here, RNA or DNA, and then the protein here, usually histone, if I'm dealing with chromatin. Now we had established that nucleic acids are made up of many nucleotides, which are linked by a particular bond. Yes, those are the kind of questions to take note of. Remember that nucleic acids are made up of nucleotides. These nucleotides are linked by a bond, and the bond is called a phosphodiester bond. Phosphodiester bond. And that bond was usually between the phosphate on the number 5 carbon atom of a particular nucleotide to the hydroxyl group on the number 3 prime. So when we looked at this, I had mentioned that ribose is labeled or is numbered. The carbons in ribose are numbered with prime. So usually, please, the link is between the hydroxyl group on 3 prime to the phosphate group on the 5 prime of another person. So you realize that that's always the link, and that's called phosphodiester bonds. So please, nucleic acid are linked by what? Nucleotides within nucleic acid are linked by phosphodiester bond. So when you want to break it, what are you using to break it? A phosphodiesterase, also called a nucleus. The nucleus name here, like I told you, was ribonucleus. So ribonucleus, that will be in RNA, then deoxyribonucleus. Now, what else can we see as a run-to question? Now, these nucleotides, when they have to be broken down, what does the nucleotide contain? It contains a sugar attached to a nitrogenous base on its first carbon, using its first carbon, the 1 prime, to attach a nitrogenous base, and using its number 5 to attach to what? A phosphate. Usually, like I taught you in our last video, it can either be 5 or 3 prime based on the situation, but usually it can enter any of them. So please, that is what we call a nucleotide. So when I want to move from nucleotide to nucleoside, what do? You get rid of the phosphate. The enzyme phosphatase adds water to remove phosphate. And once you get rid of the phosphate, what you are forming is called a nucleoside. Now, what does a nucleoside contain? Base and salt and sugar. So when you break it down, you use nucleosidases, and once they're broken down, we have our sugar moiety and our nitrogenous bases. Now, what else should we take note of? Common questions you can see in an MCQ exams, a multiple choice question exams, would be questions like, which of the following purine or pyrimidines is not found in DNA, or which of the following are found in DNA? The correct thing is that adenine, guanine, which are purines, are found both in RNA and DNA. Cytosine is found in both in RNA and DNA. Uracil is found only in RNA. Thiamine is found only in DNA. So anyhow the question is turned around, please always remember that if the question was which of the following is not found in DNA, your answer will be uracil. Which of the following is not found in RNA, your answer will be who? Thiamine. Anyhow the question is thrown at you, very easy to remember. Then remember that a pyrimidine ring is 6-membered, having 2 nitrogen and 4 carbons, whereas a purine ring is 9-membered, usually having a pyrimidine ring and an additional ring, which is an imidazole ring. Together, we observe that we have 4 nitrogens in the ring and 5 carbons. These are things to take note of. Now, when we were naming in nomenclature of nucleosides, whether purine or pyrimidine, we established that for purines, you always end with sine, but for pyrimidines, you always end with what? Dine. So when naming nucleosides, end purines with sine, end pyrimidines with what? Dines. For example, adenine will now be, after adding a sugar, it will be called adenosine. Guanine, after adding sugar, will be guanosine. Cytosine, after adding sugar, cytidine. Uracil, uridine. Thymine, you now call it thymidine. You observe the naming. Pyrimidines ends with dines. Purines, nucleotides, will always end with what? Sine. Now, this is also something that can be thrown into an exam. Very easy. Something to pass without stressing yourself. That in purine nucleosides, that the bond is always between the 1' carbon on the sugar, but it's always between the number 9, the nitrogen number 9 in the purine ring. So when you look at the ring, the number 9, that's where it will always happen. But when I have to deal with pyrimidines, 1' on the sugar, number 1 in the pyrimidine ring. So that is what to note. Additionally, while we are naming, if I'm dealing with a ribose, the name of the guy would be, you just give it adenosine as a name. You just give it cytidine as a name. You just give it guanosine as a name. But if for any reason you are using a deoxyribose, like in DNA, you always add small d before it. So small d adenosine tells me that, okay, from hearing the name ending in sine, it means that you are a purine nucleoside. The alphabet D tells me that the sugar that was used was not ribose, but what? Deoxyribose. So that is that for that. Next thing that we must note is now, in nucleotides, where is a phosphate attached? Phosphate is either to 5' or 3'. But I mentioned that if it is a ribose sugar, phosphate can also enter 2'. But usually, it's either 5 or 3'. Then, what links nucleotides? What links nucleotides together? It's always a bond called phosphodiester bond. Now, additionally, in DNA, it is not 1 strand, it is 2 strands. In DNA, it's not 1 but 2 strands. So you realize that there's a link between a nitrogenous base in one of the strands with the nitrogenous base in the other strand. There's a diagram we usually draw, that you draw a DNA following this pattern, something like this. So this link between them, nitrogenous base on one strand to the nitrogenous base on the other strand, that link is called hydrogen bond. And like I said, it's always, remember, this can only happen in DNA. RNA is single-stranded. It's DNA that is double-stranded. So in DNA, adenine will always be going to thymine, whereas guanosine, as guanine, will always be going to cytosine, guanine to cytosine, adenine to thymine. I mentioned that the link, the number of hydrogen bond between adenine and thymine is 2. Or between guanine and guanosine is how many? 3. So yeah, those are the things to note. So over here, within a particular strand, what links them is phosphodiester bond. But between 2 strands in DNA, so linking the nitrogenous base will be what? It will be hydrogen bonding. 2 between A and T, 3 between G and C. All right, so that covers our review of chemistry of nucleic acid. I hope there is no problem. There cannot be a problem. You understood very carefully. Do well to subscribe to this YouTube channel. Do well to like this video. Do well to stay connected. Comment. If you have any questions, drop it in the comment section. And surely, I am certain that you cannot have a problem with this topic again. Now, ensure you share this video to as many people as possible. Until we meet next for metabolism of nucleic acids, stay tuned to this channel. And also go through our last video. If you have not watched the video on chemistry of nucleic acids, you ought to watch it. Ensure you see the video. Thank you so much for joining us.